Imagine an immune system that is so flawlessly calibrated that it can relentlessly hunt down a microscopic, rapidly mutating virus in the dark.
Right.
It just dismantles it and then, remembers its precise chemical signature for decades. I mean, it is basically a biological superweapon.
Oh, absolutely. It's an incredibly efficient machine.
Right. But now I want you to imagine that exact same superweapon being asked to negotiate this 9-month high-stakes peace treaty with a massive, rapidly growing mass of genetically foreign tissue.
Yeah.
And that tissue is embedded directly inside its own host. I mean, that is the paradox of human pregnancy. It really shouldn't work.
It shouldn't. If you look at the biological math, the immune system should immediately identify the fetus as an invader and just, well, destroy it.
Exactly.
Yeah.
But somehow, usually it doesn't. And that's exactly what our deep dive is about today.
It is arguably the most complex immunological tightrope walk in mammalian biology. I mean, we spend so much time studying how the immune system fights that we often completely overlook the staggering complexity of how it learns to, tolerate.
Yeah. And when that tolerance fails or when it gets hijacked, the fallout doesn't just stay confined to one local area. So we are pulling together clinical immunology, neuroscience, reproductive endocrinology, and even molecular oncology today.
We are covering a lot of ground.
We really are. We're mapping out this specific devastating chain reaction, like how a failure in immune tolerance connects recurrent miscarriages, severe shifts in fetal neurodevelopment, specifically tying into the architecture of autism spectrum disorder.
Right.
And it also connects to primary ovarian failure and even post-acute neurological decay.
Which, when you look at those conditions isolated on a whiteboard, they seem entirely unrelated.
Totally. They seem like completely different worlds.
Exactly. You have reproductive failure on one end and then cognitive or neurological decline on the other. But when you start tracking the specific cellular messengers, the cytokines, the autoantibodies, the T-cell responses, this very clear unified map emerges.
A map that shows they're all connected.
Yes. The brain, the reproductive organs, the immune system, they're not operating in silos. They share a vascular and a chemical highway.
Right.
And when a massive inflammatory event causes a pileup on that highway, the damage just echoes across all three systems simultaneously.
Okay, let's unpack this. Let's start at ground zero of this immunological tightrope walk, the maternal-fetal interface. Because we generally have this cultural perception of pregnancy as this relatively passive waiting period, right?
Oh, yeah, definitely.
Like the body just sort of naturally, quietly accommodates the baby. But biochemically, maintaining a pregnancy is this aggressive, exhausting, highly active, tolerogenic process.
Active is definitely the critical word there. We have to consider the genetic reality of a fetus. It is semi-allogeneic.
Semi-allogeneic, meaning it's half formed.
Exactly. It inherits half of its human leukocyte antigens from the father. So to the maternal immune system, those paternal antigens are practically indistinguishable from a foreign pathogen.
Wow.
Or, a grafted organ. If you were to transplant a kidney with that degree of genetic mismatch into a patient, the host's T cells would just swarm and necroticize the tissue within days.
Unless you use heavy immunosuppressant drugs.
Right. Heavy drugs.
So the maternal body basically has to suppress its own baseline defensive instincts locally. But it has to do that without shutting down systemic immunity so much that the mother, comes to a common cold.
Exactly. It's a hyperlocal suppression.
And one of the primary mechanisms for maintaining this local peace treaty involves something called the TAM receptor pathway. Let's dig into the mechanics of this because it is just fascinating.
It really is.
We're talking about 3 specific tyrosine kinase receptors, TYR03, AXL, and MERTK.
Right. So these receptors, they basically act as the microscopic sanitation department at the placental boundary.
The sanitation department. I like that.
Yeah, because throughout a normal pregnancy, there is a massive amount of cellular turnover. Tissues are rapidly growing, they're remodeling, and they're dying.
Constantly.
Constantly. And when a cell dies through natural apoptosis, it has to be cleared away cleanly. That process is called efferocytosis.
Which, if I remember correctly, literally translates to carrying to the grave.
You got it. It's carrying to the grave. So when a cell undergoes this programmed cell death, it flips a specific lipid, phosphatidylserine, from the inside of its membrane to the outside.
So it's basically raising a chemical flag.
Exactly. A flag that says, hey, I am dead. Please remove me.
Right.
And the TAM receptors on the local macrophages, they recognize that flag, they bind to the dead cell, and they quietly engulf and digest it.
And if I'm tracking the biology here, the critical part isn't just that they clean up the debris, it's how they clean it up. Like, they do it silently.
Yes, the silence is everything.
Because if those dead cells aren't engulfed immediately, their membranes degrade.
Right.
And they burst open in this process called secondary necrosis.
Exactly. And when a cell bursts, it spills all of its intracellular content. So its DNA, its reactive oxygen species, its internal proteins, they all spill directly into the surrounding tissue space.
Which sounds bad.
It's very bad. The immune system immediately recognizes those spilled contents as damage-associated molecular pattern.
Like tripping an alarm system.
A massive localized alarm. It recruits inflammatory cells to the area instantly. So by efficiently clearing the apoptotic cells before they burst, the TAM receptors are actively suppressing inflammation.
They're keeping the environment quiet.
Yes, they keep the placental environment quiet and tolerogenic.
And we can see exactly how vital this silent cleanup is by looking at what happens when it, breaks down. The literature highlights these animal models utilizing triple knockout females.
Right.
So these are mice genetically engineered to completely lack TYR3, AXL, and her MERTK. And without that sanitation department, the consequences for pregnancy are just absolute.
It results in a catastrophic cascade. I mean, because the dead cells are bursting and spilling their contents, the immune system gets overwhelmed by constant inflammatory signals.
It loses its mind, basically.
It really does. It loses its ability to distinguish between self and non-self. So these triple knockout females rapidly develop a severe systemic autoimmune syndrome.
Wow.
And specifically, they begin producing massive titers of anti-phospholipid autoantibodies.
Antiphospholipid autoantibodies. Okay, we're talking about things like anti-cardiolipin, anti-phosphatidylserine, and anti-phosphatidylserine.
Yes.
And these are incredibly dangerous autoantibodies because of what they target, right? Like, we aren't talking about targeting a specific rogue protein here.
No, not at all.
Phospholipids are the fundamental structural mortar of literally every cell membrane.
They are the lipid bilayer itself. So when the maternal immune system generates autoantibodies against these structural lipids, it initiates a coordinated attack on the very foundation of the forming placenta.
The actual building blocks?
Exactly. The autoantibodies bind to the membranes of the trophoblast cells. Now, these are the cells responsible for invading the uterine and establishing blood flow.
Okay.
And this binding triggers the complement system, which physically punches holes in the cells, destroying them.
Oh, wow. And as those placental cells are destroyed, it triggers another massive systemic failure, which is the collapse of the coagulation cascade.
Yes.
The data points specifically to the thrombomodulin-protein C system, because maintaining a highly regulated anticoagulant network at that maternal-fetal boundary is, well, it's mandatory.
It is entirely mandatory. Blood flow at that boundary is highly complex.
Right.
The mother's spiral arteries have to remodel themselves to just dump high volumes of blood into the intervillous space. This bathes the fetal villi in oxygen and nutrients.
But if the inflammatory attack compromises the lining of those vessels...
Then it exposes tissue factor, and tissue factor immediately triggers clotting.
Right, and the thrombomodulin-protein C system normally acts as the brakes on that clotting process.
Yeah. Exactly. But under the sheer volume of this autoimmune attack, the brakes just burn out.
They burn out.
Yeah.
So you get widespread microthrombosis.
Yes. Countless tiny blood clots clogging up the delicate vascular networks of the placenta.
And the moment that network clots, you sever the oxygen and nutrient lifeline.
Exactly. The tissue infarctions spread rapidly and the placenta dies. This is why the vast majority of these triple knockout females suffer recurrent chronic fetal loss.
Their immune systems are systematically starving and necroticizing the pregnancy.
Yes.
It just paints this terrifying picture of cellular-friendly fire. And this targeted starvation isn't just limited to oxygen either. The research also highlights the role of folate receptor alpha autoantibodies, or FRAAs.
Right.
And the mechanism here is equally targeted and equally devastating, but it focuses on nutrient deprivation rather than vascular collapse.
Because folate is non-negotiable for early fetal development. It is required for nucleotide synthesis, meaning the fetus cannot build new DNA without it. It's particularly crucial for the closure of the neural tube in the very first weeks of gestation.
So to get that folate, the placenta expresses high levels of folate receptor alpha to capture it from the maternal bloodstream, right?
Yes, and then shuttle it across to the fetus.
But if the maternal immune system starts producing autoantibodies that structurally recognize and bind to those specific folate receptors.
It effectively blocks the loading dock.
It blocks the loading dock. That's a great way to put it.
I mean, the mother could be consuming massive amounts of dietary folate or taking high-dose supplements. Her own serum folate levels might appear completely normal on a standard blood test.
But the autoantibodies physically obstruct the receptor's binding site on the placenta.
Exactly. The folate simply bounces off. So the fetus experiences acute localized malnutrition during its absolute most vulnerable developmental window.
Which directly correlates in the clinical data with severe subfertility, recurrent miscarriage, and pregnancies complicated by neural tube defects.
It does.
So, okay, we have an immune system capable of attacking the cellular mortar, clotting the blood supply, and starving the nutrient receptors. All of this destructive potential has to be kept tightly leashed.
Yes, tightly leashed.
And that leash comes down to one fundamental power dynamic, the Th17 to Treg ratio. Because this dynamic is the engine driving almost everything we are discussing in this deep dive today.
It really is the ultimate fulcrum of immune tolerance. On one side of the scale, you have regulatory T cells, or TREGs.
The peacekeepers.
Exactly. These are the master suppressors. They secrete cytokines like IL-10 and TGF-beta, which basically tell the surrounding immune cells to stand down, stop replicating, and just ignore foreign antigens. They enforce the peace.
I always visualize this as a highly volatile diplomatic summit. Like the TREGs are the skilled diplomats, they're walking the floor, they're calming tensions, and they're actively suppressing any sudden movements.
That's a perfect analogy, because on the other side of the fulcrum, you have the Th17 cells.
Right.
These are a subset of T helper cells designed to clear mucosal pathogens, like extracellular bacteria and fungi.
And they secrete interleukin-17.
Yes, which is a highly aggressive pro-inflammatory cytokine. It recruits neutrophils and triggers massive tissue inflammation.
So in the summit analogy, the Th17 cells are the heavily armed, highly reactive castle guards.
Right.
And if the guards outnumber or outsignal the diplomats, the summit collapses into violence. A successful pregnancy absolutely requires a dominant TREG environment. The peacekeepers must remain in complete control of the local cellular environment.
And clinical immunologists are aggressively exploring how to chemically manipulate this specific ratio in patients, especially those suffering from recurrent implantation failure.
Right, where the embryo repeatedly fails to implant because the local uterine environment is too hostile.
Yes. The clinical data suggests that in those cases, the Th17 to TREG balance is heavily skewed toward the inflammatory Th17 side.
The guards are in charge.
The guards are definitely in charge. So the literature evaluates interventions using drugs like hydroxychloroquine or HCQ.
Now, HCQ has been used for decades as an antimalarial and for systemic lupus erythematosus. But its application here is fascinating because of its mechanism of action at the cellular level.
It alters the pH within the intracellular compartments called endosomes.
Endosomes.
Yes. And by changing that pH, it interferes with how antigen-presenting cells process and present targets to T cells.
Ah, I see.
The clinical outcome detailed in these studies is that HCQ administration significantly downregulates Th17-related cytokine production. It basically chemically disarms the aggressive guards.
But it doesn't just disarm the guards, right? It actively empowers the diplomats.
Yeah.
The data shows HTQ upregulates the transcription factor, FOXP3.
FOXP3 is the master regulator gene for TREG development.
Okay.
If a T cell expresses FOXP3, its absolute destiny is to become a peacekeeper. So by upregulating FOXP3 and its associated calming cytokines, the therapy attempts to forcibly rebalance the ratio. It tries to create an artificially tolerogenic environment long enough for the embryo to successfully implant and establish that vital placental connection.
It is just a remarkable piece of biochemical engineering to rescue a failing peace treaty.
It is.
But, this brings us to a much darker precipice in the data. We've established that if this TREG to Th17 balance fails in the first few weeks, the pregnancy simply ends. The embryo cannot implant or survive.
Right.
But let's move the timeline forward.
Yeah.
What happens if the pregnancy successfully establishes itself, the peace treaty holds, but then deep into the second trimester, during peak fetal brain development, that balance is violently, systematically shattered by a massive external immune event?
Then you transition from a localized failure of reproductive tolerance to a systemic catastrophic alteration of neurodevelopment.
The maternal immune system goes to war, and the fetal brain is caught in the crossfire.
Exactly. This mechanism is documented under the umbrella of maternal-to-fetal immune activation, or MIA.
MIA. And the driver of this pathology is the intrauterine cytokine storm. Let's really break down the anatomy of this storm because it dictates the entire architecture of the neurological damage that follows.
Well, a cytokine storm is basically a severe positive feedback loop.
Mm.
Usually, when a pathogen enters the body, macrophages and dendritic cells detect it and release localized cytokines to recruit help.
It's a measured response.
Right, measured.
Sure.
But in a severe acute infection, whether viral or bacterial, those initial cells panic.
Have panic.
They release overwhelming amounts of pyrogenic, highly inflammatory cytokines, primarily TNF-alpha, IL-1 beta, and crucially, interleukin-6.
IL-6. That specific cytokine is the linchpin for the cascade we were about to trace, isn't it?
It is. These cytokines completely flood the maternal serum. Now, under normal, healthy conditions, the placenta is a remarkably effective barrier.
Right. It protects the fetus from maternal hormones, toxins, most pathogens.
But it is not impervious to maternal inflammatory signaling. The sheer concentration of cytokines in a storm allows them to actually breach that placental barrier. Or, in some cases, the inflammation causes the placental cells themselves to start secreting cytokines directly into the fetal circulation.
So the maternal alarm bells cause the fetal alarm bells to start ringing deafeningly loud.
Precisely. The fetal environment, which should be deeply suppressed and quiet, suddenly becomes this bath of persistent, low-grade neuroinflammation and profound oxidative stress.
And timing is everything here.
It is.
If this storm hits during the peak windows of neocortical neurogenesis, the exact days and weeks when the fetal brain is frantically generating, migrating, and organizing billions of new neurons, the structural consequences are permanent.
The research explicitly states that this inflammatory flood alters the fetal brain transcriptome.
The transcriptome.
Yes. It lays the foundational groundwork for neurodevelopmental disorders with a very specific focus on autism spectrum disorder.
Okay, let's push on that a bit, because we know the genome is the hard-coded blueprint. When we say the transcriptome is altered by this immune activation, what is mechanically happening inside those developing fetal neurons?
It comes down to epigenetics and gene expression. The developing neural stem cells are surrounded by this soup of chemical signals that tell them exactly which genes to turn on and which to turn off to become a functioning part of the brain.
Okay.
When you introduce a flood of pro-inflammatory cytokines like IL-6 into that soup, those cytokines bind to receptors on the neural stem cells, and they activate intracellular signaling pathways like the JAK-STAT pathway that runs straight into the nucleus.
So they act as like hostile override codes.
They absolutely do. They physically alter the methylation of promoters or the modification of histones. They force the cell to misread its own instructions.
Wow.
So genes that should be promoting healthy synapse formation might be silenced, while genes promoting cellular stress responses or premature differentiation are aggressively upregulated.
You end up with a brain that is physically wired differently.
Yes. The cortical layers might organize haphazardly, or the balance between excitatory and inhibitory neurons might be permanently skewed.
And to prove that this systemic immune panic is the direct cause of these structural brain changes, the researchers utilized a very specific, highly controlled animal model using something called poly-IC.
Right.
This study is incredible because of how surgically it isolates the variable.
The poly-IC model is the gold standard for studying maternal immune activation. Poly-IC stands for polyanosinic.polycididylic acid.
Try saying that 3 times fast.
Right. It is a synthetic analog of double-stranded RNA.
Double-stranded RNA being the hallmark signature of many replicating viruses.
Exactly. Human cells don't naturally produce long chains of double-stranded RNA.
Right.
So when the immune system's toll-like receptor 3, or TLR3, detects it, the body assumes it is under a massive acute viral attack.
Okay.
By injecting pregnant murine models with poly-IC, researchers can trigger the exact physiological panic of a severe viral infection.
The fever, the cytokine storm, the immune mobilization.
Yes, all of it, but without the confounding variables of an actual live, replicating pathogen-damaging tissues itself.
Right, they isolate the pure immune response. And when they initiate this synthetic viral attack, the first thing they observe is a massive immediate spike in the mother's IL-6 levels. The primary alarm is triggered.
Yes.
And closely following that IL-6 spike, they document a sharp, systemic elevation in IL-17 in both the maternal serum and critically within the fetal compartment.
And the IL-17 elevation is where the actual tissue damage begins.
Right.
But the researchers needed to prove causality, not just correlation. So they took pregnant mice that were genetically engineered to lack the gene for IL-6 entirely. IL-6 knockout models.
Okay.
When they injected those mice with the poly-IC viral mimic, the downstream spike in IL-17 was completely abolished.
The chain reaction stalled.
It completely stalled. This proves beyond a shadow of a doubt that the maternal surge in IL-6 is the mandatory, non-negotiable upstream trigger. Without IL-6, the devastating IL-17 response simply does not occur, and the subsequent neurodevelopmental alterations in the offspring are prevented.
But when you trace this pathway in a normal immune system, you run into this bizarre, frankly terrifying phenomenon. We talked earlier about the balance between the TREGs, the diplomats, and the Th17 cells, the aggressive guards.
Yes.
The baseline assumption is that during a severe infection, the guards just naturally outmultiply the diplomats to fight the virus, right? But the cellular reality outlined here is much more sinister.
You are referring to TREG to Th17 transdifferentiation.
Right, it's an immunological betrayal.
It is a complete inversion of purpose, because key cells have a degree of plasticity. Under normal conditions, a TREG is locked into its suppressive identity by that FOXP3 transcription factor we discussed earlier.
The peacekeeper gene.
Exactly. But when a TREG is suddenly engulfed in a highly inflamed environment saturated with IL-6, the chemical pressure becomes overwhelming. The IL-6 signaling physically alters the internal programming of the peacekeeper. It forces the TREG to downregulate its FOXP3 expression and simultaneously upregulate R gamut.
And R gamma is the master transcription factor for Th17 cells.
Yes, it is.
So the diplomat literally burns their peace treaty, picks up a weapon, and transforms into an attacking guard.
That is exactly what happens.
The very cells that the body relies on to pump the brakes on the inflammation are hijacked and converted into engines that pour gasoline on the fire.
And this transdifferentiation destroys the tolerogenic environment entirely. You have a massive amplification of pathogenic Th17 effectors flooding the system, secreting IL-17, which breaches the fetal compartment.
Wow.
And this isn't just an abstract theory observed in genetically altered mice. The clinical biomarker data strongly supports this mechanism in human populations.
Right. The literature points out that elevated concentrations of IL-17 are frequently detected in the amniotic fluid of human mothers who experience severe inflammatory events during pregnancy. And crucially, those same elevated IL-17 signatures are found in the blood serum of offspring subsequently diagnosed with autism spectrum disorder. The immunological fingerprint of that cellular betrayal remains in the child's system.
It does. And the consequence of that IL-17 flooding the developing brain is what the literature classifies as fetal neuroglial toxicity.
Okay, let's break that down.
IL-17 acts as a massive accelerant for the brain's resident immune cells, the microglia.
Microglia are so fascinating because they are essentially sequestered, right? They are specialized macrophages that live exclusively within the central nervous system.
Correct. Normally, microglia exist in a ramified state. They have these long branching arms that constantly survey the surrounding brain tissue.
They're like gardeners.
Very much so. They prune weak synapses, they clear away dead neurons, and they ensure the neural circuitry is refining itself properly. They are the gardeners of the brain. But when they are bathed in IL-17 and other inflammatory cytokines from the maternal storm, they retract those long branches. They swell up into an amoeboid shape, and they become chronically aggressively activated.
The gardeners turn into exterminators.
And they get stuck in that hyper-aggressive state. They begin continuously releasing their own highly toxic pro-inflammatory mediators directly into the fragile developing neural networks.
The two primary culprits detailed here are TNF-alpha and interleukin-8.
Yes. TNF-alpha directly drives cellular apoptosis. It tells surrounding cells to die.
Right.
But IL-8 plays a very specific, highly destructive role in this context.
Because IL-8 is a powerful chemokine. Its primary biological function is to act as a homing beacon to recruit circulating neutrophils to the site of an infection.
Exactly.
But recruiting neutrophils into a developing fetal brain sounds like dropping a bomb to kill a spider.
It is an incredibly blunt instrument. Neutrophils are the first responders of the innate immune system. They are designed to swarm bacterial infections and release toxic granules full of reactive oxygen species and degradative enzymes.
Basically, to obliterate everything in the immediate vicinity.
Right. When IL-8 lures these neutrophils across the blood-brain barrier and into the fetal central nervous system, they unleash that same indiscriminate chemical warfare on the developing cortical structures. They cause profound collateral central acute phase cellular damage. The structural integrity of the brain is dismantled by the very cells trying to protect it.
Okay, so looking at this entire catastrophic chain, the altered transcriptome, the transdifferentiation of the peacekeepers, the swarming of the neutrophils, it all traces back to that initial maternal spike in IL-6.
It does.
So a logical question arises. Why hasn't human evolution simply selected against IL-6? I mean, if it is the master switch for this neurodevelopmental devastation, wait, shouldn't we view it purely as a biological villain?
That is the temptation in clinical immunology, definitely, to label a specific cytokine as purely pathogenic.
Right.
But biological systems are defined by nuance, not absolutes. If you were to completely eradicate IL-6 from a developing fetus, the results would be equally disastrous, just in a different direction.
So it's a Goldilocks mechanism.
Exactly. The literature is very careful to contextualize this. Baseline physiological levels of IL-6 are not just tolerable, they are mandatory for central nervous system development.
At those very low, tightly controlled baseline levels,
All right.
IL-6 acts as a vital neurotrophic factor.
It binds to the neural stem cells and actually guides their differentiation. It provides the necessary chemical nudges that tell a generic stem cell to mature into an astrocyte or a specific type of neuron.
It actively builds the architectural scaffolding of the brain.
Yes.
So a little IL-6 is the architect and excess of IL-6 is the wrecking ball. The pathology isn't the molecule itself. The pathology is the loss of regulation.
The immune system loses its ability to control the dose. Precisely. We are dealing with an exquisitely sensitive web of tight tolerances. When the systemic inflammation breaks those tolerances, the systems that build life pivot instantly to dismantling it.
Which provides a terrifyingly logical bridge to the next phase of this deep dive. Because we've explored how systemic inflammation alters a developing fetus and how a localized immune failure can prevent a pregnancy entirely.
Right.
But what happens when that exact same autoreactive hyperinflammatory state targets a woman's reproductive hardware long before pregnancy is even attempted?
We shift our focus from the uterus and the placenta to the primary engines of female fertility, the ovaries.
The sources establish a clear baseline here. Normal ovarian function, the maturation of follicles, the precision of steroidogenesis, the overall fertile capacity. It is heavily regulated by complex humoral and cellular immune responses.
Immune cells are always present in the ovaries, assisting with ovulation and tissue repair.
But when the tolerogenic breaks fail, the ovaries become a target for aggressive friendly fire. And this manifests clinically as autoimmune oophoreitis.
Oophoreitis refers to severe autoimmune-mediated inflammation of the ovaries.
Okay.
The disease progression is insidious. It isn't just a temporary swelling. The autoreactive immune cells chronically infiltrate the ovarian tissue, driving progressive destruction, severe atrophy, and ultimately extensive fibrosis.
So the delicate endocrine tissue is systematically replaced by rigid scar tissue.
Yes, and as the functional tissue is destroyed, the ovaries lose their ability to produce essential sex hormones and mature viable eggs.
That's devastating.
This relentless inflammatory march culminates in primary ovarian insufficiency, or POI. The patient effectively enters premature menopause, completely losing their fertile capacity, driven entirely by the collapse of immune tolerance.
And to understand how this destruction is so efficient, we have to look at exactly what the autoantibodies are targeting, because they aren't just blindly attacking the organ. Right?
No, not at all.
They are executing a highly specific strike on the core infrastructure of the ovarian follicle. The clinical data identifies high titers of pathogenic autoantibodies directed against three critical structures: the granulosa cells, the TUCA cells, and the zona pellucida.
Let's start with the zona pellucida, as it is structurally the most straightforward.
Okay.
It is the thick glycoprotein matrix that surrounds the human oocyte, the egg. It protects the egg and is absolutely essential for binding sperm during fertilization.
So if autoantibodies bind to and degrade the zona pellucida,
The egg is rendered completely non-viable.
but the attack on the granulosa and the Utica cells is arguably even more devastating because it collapses the entire endocrine foundation.
The Cuca cells and granulosa cells operate as a highly synchronized two-cell chemical factory surrounding every developing egg.
A chemical factory.
Yes, the Cuca cells form the outer layer of the follicle. Their primary job is to synthesize androgens, like testosterone, in response to pituitary signals. They then pass those androgens across the membrane to the inner layer of granulosa cells.
And the granulosa cells act as the converters. They use an enzyme called aromatase to convert those androgens directly into estrogens, primarily estriatal.
Exactly. The granulosa cells are the primary source of estrogen production in the female body, and they physically nurse the developing oocyte, providing it with vital nutrients and growth factors.
Right. So when the pathogenic autoantibodies target and destroy these specific cells, you don't just lose the egg. You completely shatter the entire steroidogenic pipeline.
The production of estrogen and progesterone crashes, the menstrual cycle violently destabilizes, and follicle development completely arrests.
But the mechanical trigger for this ovarian destruction is, well, it's one of the most bizarre and brilliant pieces of pathology in the entire outline.
It really is.
Because the attack doesn't just happen spontaneously. The immune system has to be tricked into recognizing these vital support cells as threats.
Yes.
And the molecule pulling the strings here is another cytokine, interferon gamma, or IFN gamma.
IFN gamma is primarily secreted by activated T helper 1 cells and natural killer cells. It is a potent driver of cellular immunity, usually deployed to combat intracellular pathogens like viruses.
Okay.
But when IFN gamma is chronically elevated in the ovarian environment due to systemic immune dysregulation, it executes a devastating biological hack on the granulosa cells.
This is where we need to dive into MHC molecules, the major histocompatibility complex. I generally conceptualize MHC molecules as microscopic ID badges that cells hold up to the immune system.
That's a highly accurate conceptualization. We have two primary classes. Class I-MHC is worn by almost every nucleated cell in your body.
Right.
It constantly samples the inside of the cell and holds up fragments of proteins to the immune system, essentially saying, hey, I am a healthy self cell, or I have been infected by a virus, please destroy me.
But Class II MHC is entirely different. It is a specialized, restricted badge.
Class II MHC is exclusively utilized by professional antigen-presenting cells, macrophages, dendritic cells, and B cells. The professionals. Their job is to patrol the extracellular environment, swallow up foreign debris or bacteria, break it down, and proudly display those foreign fragments on their Class II badge to passing T helper cells.
So showing a Class II badge is the physiological equivalent of handing a T cell a loaded weapon and a picture of a target.
Exactly. Which brings us back to the granulosa cells. Under normal physiological conditions, a granulosa cell is a civilian worker. It has no business interacting with T cells.
Right. It never wears a Class II badge.
Never. It is entirely focused on hormone synthesis and oocyte support.
But the research shows that high levels of IFN gamma physically force the granulosa cells to undergo aberrant MHCC2 upregulation. The cytokine forces the civilian worker to put on the uniform of a professional antigen-presenting cell.
And this is the fatal flaw in the hack. The granulosa cell now has the hardware to present targets to T cells, but because it is not a trained immune cell, it hasn't swallowed any foreign bacteria. It has nothing foreign to display.
Oh, wow.
So it simply breaks down its own internal cellular proteins, its own self-antigens, and displays them on the newly forced Class II badges.
It hands the autoreactive T cells a picture of itself.
Yes.
It's as if civilian factory workers are suddenly handed military targeting radios by IFN gamma, and they are forced to call in devastating biological airstrikes on their own factory. The immune system actively tricks the endocrine system into triggering its own autoimmune execution.
The efficiency of the destruction is just staggering, and we must recognize that this systemic inflammatory dysregulation, this sudden flood of cytokines like IFN gamma, it does not happen in a vacuum.
Yeah.
The clinical literature is intensely focused on tracking the external environmental triggers, the infections, and the profound immune stressors that can initiate these cascades and disrupt the endocrine timelines.
And as we navigate this specific portion of the clinical data, I really want to reiterate a core foundational rule of this show. We do not engage in ideological debates, and we do not take political sides. Our sole mandate is to impartially, precisely translate the documented medical literature and pharmacovigilance data provided to us, giving you, the listener, the unvarnished clinical picture of how immense immune stressors interact with the human endocrine system.
And the specific literature we are synthesizing here extensively evaluates the endocrine impacts of acute, massive immune mobilization. The sources explicitly highlight the clinical documentation surrounding SARS-CoV-2 infection and the subsequent global rollout of COVID-19 vaccination protocols,
focusing on their capacity to act as systemic stressors that can heavily alter normal female menstrual cyclicity.
Yes.
The material draws directly from official documented pharmacovigilance tracking. It specifically cites the Health Canada adverse event reports, noting page 89 of a massive 2,140-page clinical document. We're looking at documented adverse event presentations, not theoretical models.
The clinical presentation detailed in those specific reports involves young, previously healthy women presenting with severe, unresolved amenorrhea following the immune stressor of vaccination.
Amenorrhea is the complete cessation of the menstrual cycle.
Right. The clinical tracking notes patients presenting with an absence of menstruation extending for 6 to 7 weeks post-vaccination, indicating a profound temporary arrest of the hypothalamic pituitary ovarian axis we were just discussing.
The communication between the brain and the ovaries just goes completely silent. And the data notes that in severe presentations, this reproductive arrest does not occur as an isolated symptom.
It doesn't.
It is documented occurring concurrently with massive multi-organ inflammatory complications. The reports list acute kidney failure, hepatic or liver failure, and severe peripheral edema.
When you synthesize those concurrent symptoms, the liver, the kidneys, the profound vascular leakage causing the peripheral edema, what the pharmacovigilance data is documenting is a severe systemic hyperinflammatory cascade.
The immune system mounts such a massive, overwhelming response to the stressor that the collateral cytokine storm derails the tightly regulated endocrine timelines.
Exactly. The civilian factories are forced offline while the body attempts to manage the perceived systemic crisis.
The immune system rings the alarm so loud that the reproductive system just shuts down to protect itself, or it becomes collateral damage in the crossfire. It perfectly underscores the interconnected fragility of these systems.
It really does.
But this is exactly where the source material takes a wildly unexpected, almost science fiction level pivot. I mean, we've seen how reproductive organs can be targeted by a confused immune system. We've seen how a cytokine storm can alter a fetal brain.
Right.
But the final section of our deep dive unites all of these disparate threads. The brain, the ovaries, viral proteins, and systemic immune failure through a biological mechanism known as molecular mimicry.
This is the apex of the pathology. It is the specific mechanism where the immune system's remarkable ability to recognize and remember a target becomes the very instrument of its own destruction.
To understand how this connects everything, we have to introduce a new, highly specialized protein into the narrative, tenerin-4 or 10MM4.
10NM4.
This protein is encoded by the ODZ4 gene. Let's map out what 10M4 does in a healthy human body.
Well, 10MM4 is a massive, complex transmembrane protein. It spans the outer lipid membrane of the cell, with domains extending both into the extracellular space and deep into the intracellular cytoplasm.
Okay.
Its primary domain of operation is the central nervous system, where it functions essentially as the master foreman for structural neurodevelopment and repair.
The literature outlines its responsibility for regulating heavy neurological machinery. Things like axon myelination, oligodendrocyte differentiation, neurite outgrowth, and neuronal pathfinding.
So let's break those down because they represent the actual physical wiring of the brain.
Right.
Oligodendrocytes are the specialized glial cells that wrap the axons of neurons in myelin. Myelin is the lipid-rich insulation that prevents electrical signals from leaking out.
It allows for rapid, precise communication across the nervous system.
Exactly. And neuronal pathfinding is the delicate process by which a growing neuron extends its axon, navigating through a dense forest of tissue to find its precise synaptic partner.
And 10M4 is coordinating all of this movement. The text notes it specifically promotes the phosphorylation of focal adhesion kinase, or FAK.
FAK signaling is how a cell physically interacts with its environment. 10NM4 coordinates actomyosin contractility within the cell.
It literally helps the cellular membrane push outward, form protrusions, and physically reach across the microscopic gaps in the brain. It builds the bridges.
It builds the bridges. The clinical proof of its absolute necessity is observed when it malfunctions. The data shows that if you introduce a genetic knockdown or an ablation of the 10MM4 gene, FAK phosphorylation just plummets.
The oligodendrocytes fail to extend their processes.
Right. The myelin insulation stops being laid down, the structural integrity of the neural network falters, and you observe profound, measurable neurological deficit.
The text specifically highlights essential tremor as a direct clinical manifestation of 10M4 dysfunction.
So we established 10M4 as a fundamental, non-negotiable pillar of central nervous system stability. If it fails, the brain physically degrades.
But here is the extraordinary twist in the clinical data that brings us back to the reproductive system. Deep within the oncology literature, researchers discovered a direct, undeniable link between this critical brain protein and the biology of the ovaries.
Yes.
When I read this section of the sources, I literally had to stop and reread it 3 times. 10-M4 is a brain hormone. But the oncology data demonstrates a direct correlation between the downregulation of 10-M4 and significantly shorter mean survival times in patients suffering from serous carcinomas and malignant ovarian tumors.
It is a remarkable finding. If a patient's ovarian tumor tissue exhibits decreased levels of 10-M4 expression, their clinical prognosis is markedly worse.
The cancer is vastly more aggressive.
And the experimental data goes even further. When researchers actively knock down 10-M4 expression in ovarian cancer cell lines, artificially silencing the gene using targeted siRNA, those cancer cells exhibited a significant decrease in sensitivity to cisplatin.
And cisplatin is a heavy-hitting, frontline platinum-based chemotherapy drug.
By removing 10-M4, the researchers inadvertently granted the ovarian cancer cells profound drug resistance.
Wow.
This implicates 10-M4 not just as a bystander, but as a critical active regulator of cellular behavior and structural integrity within the ovarian tissue itself.
So we have a massive transmembrane protein that is absolutely vital for insulating the electrical wiring of the brain and simultaneously deeply involved in the cellular biology and regulation of the ovaries. That is the biological board we have set up.
It is.
Now we introduced the match that burns it down, the SARS-CoV-2 spike protein.
This is where we examine the mechanics of molecular mimicry. Molecular mimicry is, at its core, a case of catastrophic biological identity theft.
And the theft is exact. The source material provides a highly specific sequence located within the S1 subunit of the viral spike protein. It is a long string of amino acids, NLD, SKV, GG, NY, LY, RL, FRK, SN.
When computational immunologists analyzed that specific viral sequence, they discovered that it shares substantial structural homology with the extracellular domain of the human 10-M4 protein.
And structural homology means that when those amino acids fold into their three-dimensional shape, they look almost identical to the immune system, right?
That's right. But it gets even more precise. They zeroed in on a specific pentapeptide within that sequence, a string of exactly 5 amino acids, RLFRK.
The literature states unequivocally that this RLFRK sequence is perfectly 100% structurally identical in both the invading viral spike protein and the vital human 10-M4 protein.
It is an exact molecular doppelganger. And this is the fatal error of the hypervigilant immune system. During a severe infection or following exposure to the viral antigen, the immune system mounts a massive defense.
Right.
It utilizes those antigen-presenting cells to show the viral fragments to T cells. It creates billions of highly specialized cytotoxic T cells and neutralizing antibodies whose sole obsessive purpose is to hunt down and obliterate anything in the body displaying that specific RLFRK sequence.
The immune system builds an army of bounty hunters, and it gives every single one of them a flawless sketch of the viral suspect.
Exactly. But because of that molecular mimicry, the innocent civilian cells in the brain and the ovaries, the oligodendrocytes laying down myelin, and the structural cells of the reproductive system, they are naturally walking around displaying that exact same RLFRK sketch on their 10 and M4 proteins.
They have the same face as the virus.
Yes.
But here is the structural barrier. The brain is supposed to be a fortress. It is protected by the blood-brain barrier, a highly selective filter of tightly knit endothelial cells that prevents rogue immune cells from just wandering into the central nervous system.
Right.
For this autoimmune army to attack the brain's 10 and M4, it has to physically breach that fortress first. How does the virus facilitate that break-in?
The virus provides the biochemical crowbar. The sources detail the mechanism of the pathological breach. Elsewhere on the spike protein, separate from the mimicry sequence, there is a specific arrangement called an RGD motif, arginine glycine aspartic acid.
And what does this RGD motif do?
It binds with high affinity to integrins. Integrins are receptor proteins found on the surface of the endothelial cells that line our blood vessels. Okay, when the viral RGD motif binds to these integrins, it triggers an intracellular signaling cascade that alters the actin cytoskeleton of the endothelial cell. It forces the cell to physically contract and change shape.
And when those cells contract, they pull apart from one another.
Precisely. It unzips the tight junctions, the claudins and occludins that seal the blood vessels. This drives profound vascular leakage.
The blood-brain barrier is actively permeabilized. The fortress walls are kicked down.
And once that barrier is leaky, under the conditions of a systemic hyperinflammatory cytokine storm, the floodgates open.
Those autoreactive anti-RLFRK T cells, the bounty hunters, they just swarm out of the bloodstream and pour directly into the fragile environment of the central nervous system.
And when they arrive, they don't find the virus. They find the myelin-supporting oligodendrocytes and the delicate axonal structures expressing 10 and M4.
Oh, wow.
The T cells scan the tissue. They lock onto the perfectly matching RLFRK sequence. They assume they have found a massive viral reservoir, and they initiate a full-scale cytotoxic attack.
They release perforin to punch holes in the brain cells and granzymes to force them to commit suicide.
They slaughter the civilian workforce.
But the literature is very careful to note that this specific devastating autoimmune progression does not happen universally to every single person exposed to the antigen. The pathology requires very specific genetic hardware to execute this efficiently.
Right.
The text points to susceptible alleles, specifically highlighting HLA-DRB108.01.
This loops perfectly back to our earlier discussion about the Class II MHC ID badges. HLA-DRB1 is a gene that codes for a specific type of Class II MHC molecule.
So everyone's ID badges are shaped a little bit differently based on their genetics.
Yes, and if you happen to carry that specific AG8.01 allele, your immune system's ID badges are genetically structurally primed to bind to and present that exact RLFRK sequence to your T cells with terrifying efficiency.
Your body is just genetically superior at amplifying the sketch of the suspect.
Which, in the cruel irony of molecular mimicry, makes the resulting autoimmune attack exponentially more devastating.
Which brings us to the ultimate, horrifying synthesis of everything we've discussed today. We have an army of autoreactive T cells slaughtering the oligodendrocytes in the brain, driven by an exact genetic misidentification, flooding through a chemically compromised blood-brain barrier.
Yes.
Why doesn't the immune system eventually realize its mistake? Where are the safety mechanisms? Where are the TREGs, the diplomats, to shut this down?
They are gone.
Gone.
If we connect this terminal phase back to the mechanisms of the intrabotulinum cytokine storm we discussed earlier,
The TREG to T817 transdifferentiation.
that is the final fatal blow. Because this entire cascade is occurring under severe systemic viral inflammation, the overwhelming presence of cytokines like IL-6 has already forced the regulatory T cells to transdifferentiate.
The diplomats have been biochemically reprogrammed into pro-inflammatory T817 assassins.
There is literally no one left to call a ceasefire. The text explicitly states this cascade proceeds unchecked by immunological tolerogenic systems. The brakes on the immune system have been completely physically removed.
And the clinical result of that unchecked autoimmune rampage is the profound pathology observed in post-acute neurological sequelae.
The clinical data documents widespread axonal dystrophy. The electrical wires fray.
You see profound demyelination because the oligodendrocytes that lay down the insulation have been systematically eradicated.
And the final piece of the structural collapse is the decay of vital synaptic proteins. The text lists the destruction of synaptophysin and PSD95,
which are the fundamental structural proteins that allow neurons to physically connect and communicate with one another.
The network isn't just inflamed, it is being actively dismantled by the very superweapon designed to protect it.
It is the ultimate manifestation of immune dysregulation.
It really is.
So we have covered a staggering complex landscape of human biology in this deep dive today.
We certainly have.
We started by examining the razor's edge of early pregnancy, detailing how a flawlessly executed dance of immune tolerance, managed by the silent cleanup of TAM receptors and the delicate balance of diplomats and guards, is the only thing standing between a developing life and immediate autoimmune rejection.
Right.
We then traced how, if a cytokine storm hits during gestation, the resulting flood of IL-6 and IL-17 can violently alter a fetal brain's genetic blueprint, transdifferentiating the immune system's peacekeepers and unleashing neuroglial toxicity that lays the architecture for autism spectrum disorder.
We followed that same autoreactive pathology right into the reproductive hardware, breaking down how high levels of IFN gamma can execute a biological hack, forcing civilian ovarian granulosa cells to present their own antigens,
triggering autoimmunophoritis and the collapse of the endocrine system into primary ovarian insufficiency.
Exactly.
And finally, we decoded the terrifying reality of molecular mimicry. We saw how a tiny 5-amino acid sequence, RLFRK, can perfectly trick the immune system into confusing the vital 10M4 brain and ovarian protein for a viral invader.
We watched the virus pry open the blood-brain barrier, allowing a genetically primed, unchecked army of T cells to dismantle the nervous system while the regulatory brakes were completely offline.
Why does walking through this intense microscopic detail matter to you? It matters because it shatters the illusion that our bodies operate as isolated silos. A respiratory virus, an autoimmune flare-up, a shift in reproductive hormones, and a decline in cognitive function are not separate files in a medical cabinet.
They are inextricably linked, speaking to each other across a vast, complex chemical highway.
And acknowledging that systemic crosstalk is the only way modern medicine can hope to transition from merely managing isolated symptoms to actually halting the underlying interconnected engines of chronic disease.
Which is exactly what the future of immunology is working toward.
And that search for a way to halt the engine brings us to the final, incredibly provocative note left in the source material. We've spent this entire deep dive analyzing how the STAT3 and IL-6 axis acts as the master switch, driving this devastating hyperinflammatory neuro-ovarian feedback loop. But if we can map the exact wiring of that switch, it means we can design a way to turn it off.
Yes.
The literature points toward a fascinating frontier, repurposing existing classes of immunotherapeutic drugs, specifically selective estrogen receptor modulators, or SERMs. The text highlights a drug called basedoxifene.
Basedoxifene.
Yes, the hypothesis is that if molecules like basedoxifene can be leveraged to precisely, selectively downregulate that hyperactive STAT3 IL-6 axis, we might possess the pharmacological tools to step in and forcefully hit the brakes.
We might be able to artificially rescue those ablated neuroovarian pathways, forcing the transdifferentiated T cells to lay down their weapons and rewriting the peace treaty before the permanent structural damage is done.
It is not a finalized cure, but it is a massive, tangible glimmer of hope at the bleeding edge of immunology. It means the system can be repaired. And that is a thought I want to leave you with to ponder on your own. Thank you for joining us on this incredibly dense, fascinating journey. When we started, we talked about the comfort of an X-ray, the desire for a clean, simple, visible answer.
But the reality of your biology is infinitely more complex and, frankly, infinitely more beautiful than a black and white photograph.
You are a walking, breathing, highly negotiated ecosystem. The diagnostic waters might be murky, but as we've explored today, if you look close enough at the microscopic machinery, the patterns are there, waiting to be understood. Stay curious.